Hello students, welcome back. We're going to continue today with Carbohydrates Part 11. Carbohydrates Part 11. Let us continue. We've talked about all of our monosaccharides and our monosaccharide derivatives. We've talked about our disaccharides. We've talked about the disaccharides. In this lecture, we're going to start talking about our polymers. And there are two special types of polymers that have repeating disaccharide units. And let's talk about those next. Okay, the first thing we're going to do is create the disaccharide unit. So we're going to have here, we're going to make glucose. You all are going to get tired of drawing glucose. So that's down, up, down, and then we'll make it alpha. Now, normally on glucose, don't draw this because I'm going to erase it as soon as I draw it. If we were just going to write glucose, we would put CH2OH. But we aren't going to have the alcohol. We're going to oxidize that alcohol. That's the, okay, what carbon? That's carbon 1, 2, 3, 4, 5, 6. So this is the number 6 carbon. What we're going to do, we're going to completely oxidize this number 6 carbon to COO. Well, let's just put COOH for right now. Okay. We completely oxidize that number 6 carbon of an aldehyde, of an aldose. Let's see if anyone remembers. What's the name of an aldose when you oxidize carbon 6 to a carboxylic acid? Exactly, uronic acid. So this would be glucuronic acid, but we aren't going to have glucuronic acid. We're going to lose the proton, and we're going to have just its conjugate base. So this is no longer glucuronic acid. It's glucuronate. Glucuronate. Okay. So what we're trying to do is create a disaccharide. It's going to be a repeating unit. So this is glucuronate. I'm going to make this a little smaller. Okay. And now let's make ourselves a second sugar. And this second sugar is going to be, it's going to be an amino sugar. And we've seen the amino sugars already. Okay, it's going to be a derivative of glucose. Lots of these sugars are derivatives of glucose. And so up top, we're going to make a sulfate. So we're going to have a sulfate up top on carbon 6. And on carbon 2, since it's an amino sugar, we have to have an amine on carbon 6 and a sulfo group, SO3 minus. And the rest of it is just glucose. And we'll make this alpha, it doesn't matter. And so this compound, I did not write it, and I need to have written it. But I'm going out of order, and I didn't write the name of this one, but that's okay. The name of this compound is it's going to be N-sulfo 6-glucosamine sulfate. Okay, I'm going to highlight the sulfo group, the SO3, instead of SO4. That's the sulfo group in yellow, and in blue here, this is the sulfate. So if we number these 1, 2, 3, 4, 5, and 6. Right, the one on top is the sulfate. Sulfates are SO4s. The sulfo here in this context is going to be an SO3. this a little smaller. And so those are our two sugars that we're going to put together to make our disaccharide unit. So basically what's going to happen, and again, it's going to be a glycosidic bond. So what's going to happen, this O here, the 4O is going to attack the anomeric carbon. And now I'm going to just draw the product. And you know what this is, but I'll tell you, you'll find out in just a second. So I'm going to draw my glucuronate here. Okay, that doesn't want to work. I'll make my glucuronate red. Down, up, down. Now, well, I'm going to draw my other one first before I make the bond. CH2OSO3 minus OH, and then this is NH SO3 minus up. And the only thing now, I've drawn both of them. I just haven't put the bond in between them. Now, I'm going to ask you all a question. Does everyone see there's a vertex right here? See where this corner is for that corner? I didn't put a letter there, but what does that corner represent? Can anybody tell me what these corners represent? What atom do they represent? They represent carbons. So in organic chemistry, or in chemistry, whenever you have a corner, when you're making a compound and there's a corner or a vertex, that represents carbon. Like if I drew this, let's see, that's 1, 2, 3, 4, 5, 6. That represents 6 carbons, so this would be hexane, right? Because it's just six points. Make sense? Now, that's always true except for one situation. That's when you're making a glycosidic bond. Now, what I want to do here, this has to be an alpha linkage between this carbon here, between this carbon and this carbon, an alpha linkage. And so, what I'm going to do here, I'm going to draw a vertex here, go over, put my O, go over, and go up. And so there's an alpha 1,4 bond, but it has those corners. But in a glycosidic bond, the corners are not carbons. They are not carbons. So in a glycosidic bond, the corners, that's the only time, well, that's one of the only times in chemistry where a vertex is not a carbon. Is that clear? Because you'll see that a lot. And the reason you do those, the reason we make these corners in glycosidic bonds is because sometimes they go up and the other one goes down. It's just easier to draw these vertices this way. Is that clear? Does that rationale make sense? So just remember here that these are, I'm going to put corners are not carbons. They're nothing. They're just a way to get the bond from the anomeric carbon to whatever the other carbon is. You just want to get that glycosidic bond from one carbon to another one, and those corners are just the way to do it. Now, I could do it like this. They could have scientists could have come up with this convention. They could have said, well, instead of doing corners, let's just do a smooth curve. Let's just do it this way. Make it curve from here to here, and there's no corner. And they could have done it that way, but that's not how it's. Typically, it's not drawn that way. Typically, it's drawn this way, and you'll have corners. Okay, makes sense? Now, this compound is a repeating disaccharide. I'm going to put here, and so this is going to repeat. I'm going to put our brackets here, and I'm going to put an N here. And this repeating disaccharide unit is a polymer. And so we call this repeating disaccharide unit that has, over here we just have a sugar, and so this is our glycose. The first one is always a glycose, just a sugar, some sort of sugar. It could be any sugar, the first one. But the second one is an aminoglycose. The second one is always an aminoglycose. See, the first one only has a glycose unit. It's just a glycosyl unit. But that second one is always an aminoglycose. And so whenever you see this repeating unit of glycose, aminoglycose, glycose, aminoglycose, glycose, aminoglycose, we call that, and it's a long polymer, we call that long polymer, it has a special name. It's called a glycose aminoglycan. A glycose aminoglycan is the name of this repeating unit of glycose, aminoglycose, glycose, aminoglycose. And these are very important in biochemistry. They're very important in biochemistry. These glycose aminoglycans, they're part of these membrane proteins, or they're part of proteins called proteoglycans. And I'm just going to put here part of proteoglycans. So they are part of, and we'll talk about proteoglycans next. I mean, at some point, not next. Okay. One of you is going to be able to figure out that's part of proteoglycan. I'm sorry. Proteoglycan. A proteoglycan is made up of two macromolecules. Two macromolecules. Can anybody tell me what those two macromolecules are that make up a proteoglycan? You have to know it. What was that? A protein and a sugar. A protein and a sugar. A proteo is for protein. Glycan is for the glycose, or the sugar, or the carbohydrate. Okay. Now, this proteoglycan that we've made here is a specific proteoglycan. And you all already know what it is, but you just didn't know what it looked like. Okay. First, I'm going to ask, can anybody recognize it from just its structure? Chances are you could. But let me give you a hint. This polymer can prevent blood from clotting. It prevents blood from clotting. No. Sometimes when you're in a hospital and they take your blood, this is already in the tube so your blood won't clot. What's the name of that polymer? What's the name of that carbohydrate? What's the name of that glycosaminoglycan that's in your blood and it prevents your blood from clotting? No. No, I'm surprised no one knows it. I'm surprised. Okay, you want me to give you the first letter? Okay, the first letter is H. I'm surprised. E. P. A. Heparin, exactly. Heparin. Heparin. Yeah, you all would have been dead on hangman. But yes, that's heparin. So heparin is a glycosaminoglycan. And what they do in hospitals, they'll already have tubes that have heparin in them. And so what they'll do, they'll put the blood in the heparinized tubes, and it prevents your blood from clotting so they can do whatever tests, whatever lab tests they need to do on the blood. And heparin is a glycosaminoglycan. There are several glycosaminoglycans, but we're only going to go over two of them. Only 2 of them, and that's one of them. And so it's a repeating disaccharide unit. Okay, is that clear? Everyone okay with that one? We're only going to do those 2. We haven't gone over the next one. We're ready to go over the second one now. Yes. So we've done heparin. That's one glycosaminoglycan. We're going to do one more. Okay. Everyone okay with this? Okay. Let's go to our second glycosaminoglycan. Okay. Okay. Let's see here. Okay, I'm going to draw a structure here, and I want you all to tell me what it is. Okay, this is going to be the first molecule. Well, I'm going to wait here. Up, down, up, down, and this is going to be up. Okay, and I'm going to do this here. Okay. Can anyone name this structure? Glucuronate. Alpha or beta? So this is beta glucuronate. Why is it not glucuronic acid? Why is this not glucuronic acid? Exactly, because we've lost the proton. It's because you need to be an acid, you have to have a proton, and this has lost its proton. So this is the conjugate base of glucuronic acid. Okay, and so that's going to be our glycose unit. Now let's look at our second one. Okay, we're gonna develop this one with you here. Now I'm going to go, this is going to be CH2OH. This is up. And the second one is going to be up. And then the rest of it is just going to be just like glucose. Okay. I'm going to erase this in just a second, so you may not want to write that part. I'm going to erase the circled one in just a second. Okay, but I want to see if anyone, so this is let's see this is an epimer of glucose. 1, 2, 3, 4, 5, 6. What number epimer is this of glucose? This is the 4 epimer of glucose. What molecule or what carbohydrate is the 4 epimer of glucose? What molecule is the 4 epimer of glucose? Not mannose. Maltose is 2. It's going to be galactose. I'm just going to put galact here because we have to name this. So this would be galactose. And so now I'm going to be asking you all a lot of questions today. You're going to like this. I'm going to write something here. I'm going to keep the part. Whatever I put in red, I'm going to erase eventually. So be ready for it. So I'm going to put here N, H, and then I'm going to put here H. So we put an amino group onto galactose. We put an amino group onto galactose. What is the name of this sugar derivative if you put an amino group onto galactose? Galactosamine, exactly. And so this is galactose. This is galactosamine. Now I'm going to do one more thing to this. I'm going to erase this, and nothing is going to be red now because we're going to keep everything. Now I'm going to put an acetyl group. An acetyl group here. This is an acetyl group. And so this is acetyl, A-C-E-T-Y-L. is that's green. Now we've put an acetyl group onto galactosamine. Okay? What is the name of the carbohydrate derivative when you put an acetyl group onto galactosamine? N-acetyl galactosamine. Exactly. And so, because it went on to the N, the carbon, so this compound is N-acetyl galactosamine. So on the left we have our glycose, and on the right we have our aminoglycose. So this is an aminoglycose. Okay. Now we're going to put the two of these together. Okay. Now this is going to be different. We've never seen this one. It's going to be the 3, in this situation, it's going to be the 3-hydroxyl is going to attack the anomeric carbon. The 3-hydroxyl. Okay. And so this is what we're going to end up with. COO minus, down, up, down. I'm not going to do anything to the anomeric carbon yet until we make the bond. And now for the aminoglycan, CH2OH, and this one is going to go up. And this, I'm not going to do anything to the 3 yet. And then NH, C double bond O, CH3, and this goes, well actually, I'm going to erase these N ones right now because they're going to do something special. So there's an H here, and this is going to go up, O, and an H here. Okay, now we're going to make the bond. The bond is going to be between the 1 on the left and the 3 on the right. 4, 5, and 6. Okay. So now let's make this compound. So what's going to happen, this first bond is going to be beta, so this has to go up to the O, and then this one is going to go down here. Now these bends here, these corners that I have in this bond here, are these carbons? No, they're not. It's just a way to show you, just to get from one carbon to another carbon. But the main thing here is that this one has to be up to make this beta, and this has to be up to make this galactose. No, we started with beta. We started out with beta. Right. If we started out, if we wanted this to be alpha, then this bond would have to go. I'm just going to erase this for just a second to show you this. If we started out, if we wanted to make an alpha linkage, what we have to do, we'd have to go down here. This would go down. And then we need to go, this would go down from here. And this one needs to go up. And so we just need to connect them some kind of way. But we don't want alpha. So I'm going to erase this. So we want beta, so this has to go up. And the key here, the main thing you want to remember is that these corners, In a glycosidic bond, those corners, they are not carbons. They're just a way to get the bond from one carbon to another carbon. Everyone okay with that? Okay, put your notes away. Oh, questions first. N-acetylgalactosamine. I would probably develop this slowly doing the test. For example, you know what glucose is. You know what glucose looks like. And if on the test, I would just show glucose and I would add this. And I would say, what's the name of this structure? And you'd have to be able to say, oh, this is glucuronic. Or if I had the H there, you'd say glucuronic acid. Or I would ask you, I could put galactose and put it there. Because I need for you to know on the test, I need you to know there are 3 sugars that you have to know for the test. We're going to go over this. You have to know what galactose is. You have to know what sugar. You have to know what fructose is. And then those really are the only 2 you need to just know outright. Because everything else, like I would say to you on the test, I would say, well, know that galactose is a 4 epimer. That's probably the one I know. I want you to know. Those 3. Glucose, fructose, you just have to know those structures purely. And the third one is going to be galactose. But all you have to remember is that it's the 4 epimer of glucose. And anything else I asked you to draw, you could know. It would be based on glucose. Like I could ask you to test, draw for me the 3 epimer of glucose in the Hayward projection or in the Fisher projection. And so you have to know what glucose looks like first, and then you can easily draw that. Question?
What is that glycosaminoglycan?
Oh, yes, I'm giving you the name of this one. Yes. This one, now this one is found in tendons, in ligaments, and it's found in cartilage. And this glycosaminoglycan is known as chondroitin sulfate. Not as famous as heparin. Chondroitin sulfate. So remember, connective tissues have a matrix, an extracellular matrix, and these glycosaminoglycans are found heavily in that extracellular matrix, things like chondroitin sulfate. And they're also part of the proteoglycans. Well, all of the glycosaminoglycans are part of the proteoglycans.
And this is for repeating disaccharides?
Yes, that's right. I didn't show you that. Yes, exactly. Let me show you where it repeats. So what's going to happen here? You're going to have here and here and in. So that's just you're going to keep repeating those disaccharide units, and that's going to make chondroitin sulfate. And so there are several glycosaminoglycans, but those are the 2 most common ones that you'll see in medicine that are related to medicine.
And this is part of the connective tissue.
Right, it's found in cartilage, tendons, and ligaments, things like that. Lots of connective tissue have these chondroitin sulfates. But these glycosaminoglycans in general are found in proteoglycans, and that's what we'll talk about next time.
And the same thing about the corners, the anomeric carbon, and the one bonds that makes the glycidic bond on this one as well are not.
Yes, the corners again. The corners on a glycosidic bond, corners are not carbons. That's very. Yes, if there's a glycosidic bond, those corners are not carbons. Ever. Question?
When you say like 4 epimer or 2 epimer, is that where to change them like 4 carbons?
Exactly. Just like for example, glucose, if you go from 2 to 5, right, left, right, right. If it's a 4 epimer of glucose, so that would be 2 would be right, left. So the 4 epimer would be right, left, left, right. You just change 4 around. You just flip 4. Carbon number 4. That would be galactose. The 4-epimer glucose is galactose. If you change carbon number 4. Right. So D-glucose is not an epimer. It is a diastereomer. Exactly. It's a diastereomer. What if I change, what if instead of having right, left, right, what if I had left, right, left? What would that be called? L-glucose. Exactly. It's the complete mirror image. And so that's not an epimer or a diastereomer. What is it called if you flip all of the chiral centers over? No? An enantiomer. Exactly. An enantiomer. You flip them all over. Exactly. I only showed you one example, and it is the 4-epimer. But I can ask you, draw me the 2-epimer, and you just flip 2 around. Or, oh, you don't have to worry about the name. Yeah, I don't care about the name. I would just ask you to draw it. Make sense?
Yeah, I do have a question. In one of the videos, you were saying, I'm trying to find where it's at. I think it was on the muramic acid. You were saying that it was on the 4th carbon, and the thing that you were drawing it on the 3rd carbon. So did you mean the 3rd carbon, or what was it?
Yes.
It was on the alpha D glucosamine with the lactic acid?
Yes, I did say the wrong carbon.
You said the 4th.
I said but it was supposed to be on.
Drawn it on the 3rd.
Yeah, it's the 3rd. It's
So
the,
the
yeah.
3rd is the
I said the wrong, yeah, because I can remember seeing that structure.
That was the only
Yes.
when I was watching the video.
Yes.
I was like,
Yeah, muramic acid, you're putting, I think it's a lactose. Yes, you're putting the lactose on, you're supposed to put the lactose on carbon number 3, not carbon number 4. That's muramic
it
acid.
was the 3rd amino sugar on part 7.
Yeah, that was muramic acid. Yeah, that's carbon 3, not carbon 4. Okay, I guess I won't ask you all any questions today since you all have asked so many. Have a great day.